Good afternoon. Welcome to the PROMETRIKA webinar series, Keys to a Successful Natural History Study. Today's session is entitled A Statistical Problem at its Core. My name is Megan Stepanius. I'm the president, founder, CEO of PROMETRIKA, and I'm a biostatistician, a PhD biostatistician with more than 30 years of experience in clinical research. Today, I'm very pleased to be joined by our guest, Dr. Jinma Ran. Jinma is the Director of Statistics supporting oncology and health economics and outcomes research at Pfizer. I'm also joined by my colleague, Suzanne Granger. Suzanne is a Senior Manager of Biostatistics at PROMETRIKA, and she has more than 25 years of clinical research experience. As I said, this is the third of our three-part series of webinars, and I will quickly recap part one and part two, and then we will delve into the statistical aspects of natural history studies. This topic will be divided into the natural history data's role in clinical endpoint development and in surrogate endpoint development. And there will be a great deal of focus on natural history data as an external control. There will be a comprehensive review of design and analysis considerations presented by our guest, Dr. Jinma Ran, and a case study of a retrospective natural history data collection that was planned to be used as an external control, and that will be presented by my colleague, Suzanne. A quick look at parts one and two. We covered the various types of natural history studies and talked about the spectrum and how what's the level of natural history data collection for these different types along the spectrum, starting from retrospective natural history studies that are almost completely made up of natural history data, registries similarly, and going to randomized clinical trials that have parts of them collecting natural history data, such as medical history or randomized clinical trials that borrow from natural history data in their analyses. Our clinical operations colleagues covered different pitfalls of running prospective and retrospective natural history studies and what are the considerations for a successful conduct of a natural history study. And they delved into fit-for-purpose clinical operation processes for natural history studies. Our data management colleagues covered CRF and CCG design for natural history studies, and they talked about the requirements for databases that collect natural history studies, including simplicity, flexibility, and how time points and so-called visits are structured in natural history data collection. Finally, in one of these two sessions, we covered in depth a case study where one of our sponsors had conducted a prospective natural history study that was disrupted by COVID-19 and resulted in a significant reduction of the data that was collected. The sponsor collaborated with an academic investigator to supplement the data, reimagine the study objectives, and finally achieve the goals of the natural history study by applying creative statistical methods and addressed additionally the practical and data challenges in these types of studies. So now our deep dive into the statistical considerations for natural history studies. One of the most common usages of natural history data is clinical outcomes development. What are clinical outcomes? Clinical outcomes are outcomes that directly measure if a patient feels and functions better or lives longer. Natural history data are used in clinical outcome development, um, and they are instrumental in sponsors deciding on primary and secondary efficacy endpoints for their randomized clinical trials, and on deciding on the timing of those measurements for their randomized clinical trials. Natural history data is also very commonly used to derive sample size estimation assumptions for variability and potential effect size for clinical outcomes in order to estimate the sample size that's needed for a randomized clinical trial. Natural history data are also used for development of surrogate endpoints. What are surrogate endpoints? Surrogate endpoints are those that do not directly measure clinical benefit, but can predict clinical benefit. Surrogate endpoints can be used instead of clinical outcomes in situations in clinical development programs where it can be shown that clinical outcomes take a very long time to study, and they may take a very large sample size to study. And there may be other reasons of lack of feasibility of having a clinical endpoint as your primary endpoint for a trial. Natural history data can be used in this case to evaluate whether surrogate endpoints are predictive of clinical endpoints. So they are used to evaluate the predictive nature of the surrogate endpoint. So we come across in our practice many sponsors who are interested in bringing to regulators a novel surrogate endpoint, and in developing a novel surrogate endpoint in order for it to be acceptable to regulators as a reasonably likely surrogate endpoint. And in order for that endpoint to be used as a accelerated approval endpoint for FDA's accelerated approval pathway, whereby a short-term study can be used for a conditional approval and marketing of a product, and then a post-marketing study can be used to show the efficacy in the long term and with greater numbers with respect to the clinical endpoint, and also to show the relationship between the surrogate endpoint and the clinical endpoint. So reasonably likely surrogate endpoints that can serve this type of a clinical development plan are those that have biological feasibility. In other words, there's a biological relationship that supports that surrogate endpoint's ability to predict clinical benefit, and that they have and that there is a lack of clinical data, sufficient clinical data to consider that endpoint a validated surrogate endpoint. So there's a distinction between a reasonably likely surrogate endpoint and a fully validated surrogate endpoint. But the reasonably likely surrogate endpoints, they can be used for the accelerated approval pathway. So natural history studies are very much used to develop these surrogate endpoints. And what are the natural history studies that are fit for purpose for this exercise? Well, they should have sufficient sample size to support the model building. In rare diseases, sometimes we end up combining studies in order to achieve this, and they should have sufficient length of follow-up, so that the change in surrogate endpoint is observed, and the change in the same patient in the relevant clinical outcome is also observed in the natural history study. These studies, in order to be fit for purpose for surrogate endpoint development, should also have a rich data set of the confounding variables that need to be controlled in order to show the relationship between a surrogate endpoint and a clinical outcome. So how is this done? It's basically model building, and building models and different types of models with, depending on the type of data, but building models that predict a clinical outcome using a surrogate endpoint, and use the natural history data to do it. So in these models, the dependent variable is the change in clinical outcome over a clinically relevant time frame. The predictive variable is the change in the surrogate endpoint, again, over a reasonable time frame. And the model would control for all measured potential confounders. And the usual model building tools are used to optimize the model. And again, the goal is to show that a change in surrogate over a practical time frame predicts a relevant change in clinical outcome. An example of this is some work that we did in a rare disease population of rare kidney disease. And the clinical endpoint was a change in eGFR over 2 years, the clinical endpoint of interest. The surrogate that was used was a change in proteinuria over a shorter time frame, let's say 6 months. And the model used natural history data from several sources because of the rare nature of this disease, combining all those data sets. Fortunately, there was this rich set of data that included measurements of potential confounders. So the models that were built controlled for potential confounders. And at the end, it was successfully shown that a pre-specified change in surrogate endpoint predicts a relevant change in clinical outcome. I now turn it over to Jinma to cover design and analysis of externally controlled studies. Jinma?
Thank you, Mignash, for the introduction and the great presentation. From your presentation, we already learned how to develop the endpoints, the surrogate endpoints. Now let's talk about the design and analysis of externally controlled studies. My colleague Joe Capillari is also the co-author on this topic. I do not have any conflict of interest to disclose. I will try to cover the 5 items in list here. Probably I cannot, for some items, I might just touch on a little bit because the time is limited. Let's just quickly review the regulatory guidance. In the past 20 years, like 20 years ago, ICH E10, they already mentioned using external control, but they already discussed the main advantage and the disadvantages. And recently in EMA adopted reflection paper, they also mentioned that the formal incorporation of historical controls with concurrent control data is possible, but inherently introduces further uncertainties to such comparison. Recently, FDA also issued some guidances. So in 2019, the FDA guidance for rare disease using natural history study in rare disease. They also add additional considerations for using external controls. And just last month, FDA also issued the real-world evidence guidance. Have the consideration for the using real-world evidence or real-world data in both interventional and non-interventional studies. As we all know, the external control, historical control is part of the real-world data. When we're using external control design, well, think about this is one, it will be more persuasive. It's acceptable for regulatory bodies. I summarize a few bullets here. The use of external control design is most persuasive when meet those criteria. There's no feasible placebo control. It's no available therapy for comparisons. The disease progression is well understood or predictable. So the outcome measure is objective with a large treatment effect. And the population and the setting between this trial and the external control is similar. So if you want to know details, you can read that paper I listed here. As we all know, the external control has been widely used. It's especially for supporting the single-arm trial, the supplement single-arm trial, but it's also sometimes support the RCT. The single-arm trial it's common use in oncology and rare disease. On the left side, I'll give you just an example that the single-arm trial has become used in FDA initial accelerated approval indications in malignant hematology and oncology. Then on the other hand, on the right side, will show that the single-arm trial for the FDA submissions, but their external comparatives is commonly used to support this FDA submissions too. It's the top 5 HTA bodies. On the brown colors shows using real-world data for the external comparators. As we all know, that's real external control, external historical control is part of real-world data. Okay, now let's talk about the core, how to design and analyze those external control studies. Here's a 4-step approach discussed based on gray paper. So the first step is to assess the feasibility of data source and the study design. The step 2, we need to adjust those baseline characteristics and the potential confounders. The step 3, we could use quantitative bias analysis to assess the threat of bias. And the final step, step 4, is combine the outcomes through this Bayesian borrowing approach. Just need to be noted that this 4-step approach it broadly covers all external control studies, including the supplemented single-arm trial and augmented RCT. The step 4 is applicable only to augmented RCTs, not a single-arm trial. So it's not necessary always need a step 4. The dotted line from step 3 indicates that this step may be reordered. And another way to say the step 3 is not always a step 3. There could be changed to step 2. It depends on the situation. Okay, the first step, when we want to design the external control studies, we need to think about the first question is, do we really need external control or not? Here, just give an example how to make the decision. We need to think about a couple of these factors. The first, do we have the quality historical control data? If we do not have the data, it's not mentioned to use it. Is that using RCT is the ethical? Do you have the recruitment challenge using RCT? Is this data for the confirmatory study or not? A couple of the items, there's the factors, the key factors you need to think about before you make a decision using historical control. Okay, if you want more details, you can read this paper. I'm sorry, this word may be too small, the size cannot read clearly. Once we decide to use the external control, then we need to apply some criteria to select external control. Here's a Pocock's criteria, including the 6 items here. The first is subject to same eligibility criteria, trying to reduce the selection bias. So then when you think about the distribution of the important criteria characteristics to reduce the confounding bias, information bias, and to think about identical treatment, reduce the potential, the positive treatment bias, the treatment outcomes evaluated in the same manner, reduce the information bias. And we also consider that's collected recently in the same setting by the same investigators, all trying to reduce those other potential confounders. The most common type of study for using external control is observational or interventional. We all know that we assign the patients to receive an intervention or treatment or not. So then, if, for example, if we have a single arm trial, then we want to use the external control. There could be it's retrospective or prospective. Use the historical control as a retrospective study because this the data collected after the outcome observed. It could be it's a prospective is it's a longitudinal cohort. The data collected before this outcome observed. Another very important thing we need to think about is here is we need to identify those key factors, those affecting bias. We can have a literature review. We could collect the expert opinion, all based on the previous data, with the purpose of trying to collect all potential confounders. But it's not easy to include everything, right? All potential confounders. We could have unmade confounders. It's okay. We will try to show our effort, try our best to include those main confounders. And we also could use some covariates that are associated with unmade confounders. We're trying to reduce those bias. Once we have the study design, we collect those data, then we need to apply some statistical methods to adjust those confounders or reduce this potential confounding. Here it's list a couple of methods here for frequentist. If we have the individual data, we could apply the G computation. We could use propensity score-based weighting methods. The most common is that IPTW. Now, you also could use OW, SMR, just here. We also could use doubly robust estimator, the kind of combined G computation and the IPTW. And we also could use matching or stratification method. If we only have summary data, sometimes we do not have individual patient level data. We also could use network analysis. We use simulated treatment outcome or matching adjusted indirect comparison. For Bayesian, most of the Bayesian, they also have MAC or MAP could use. It's not common use here, but I just list a couple here for your reference. The third step, we could use quantitative bias analysis to assess the threat of bias. We already done the analysis. Then we just think about this. Do we need another sensitivity analysis? Sometimes we do need. For example, we have the parameter, the age, gender, or something that's in the model. So we could set up this like age is 50 years old, it's the average. How about that's 40 years old? How about 60 years old? It's any big change on the treatment, on outcome of treatment effect, right? We could, at the same time, we also can change, put this different, multiple parameters, later, like age, gender, or it's even the race, all together. We could set up, change the value to see how impact on the outcome. And we also put the distribution, probability distribution on those parameters. So these are all kind of sensitivity analysis will help us to assess the threat of bias. Some people also like to report the e-value. This indicates how strong a measured confounder is needed to fully explain away the treatment effect. Okay, this is this is just a sensitivity analysis to help us assess the threat of the bias for the step 3. Then the final step is called the Bayesian dynamic borrowing. As I mentioned before, this is not always needed. So this step helps to create the priors from external comparative data. So the external comparator could be from one source, like the graph A. It could be have multiple sources, the external control. So then you could use the meta-analysis model to get MAP, because prior. So which all can help us to just apply to the internal control data to increase the total power of the control group. So again, this is only be applied to augmented RCT, not to supplemented single arm trial. It's not common use, but it's give you one of the options. Okay, let's quickly summarize. So in study design first, we need to think about the external control design is most persuasive when they meet those criteria already mentioned before. During data collection, we need to try to include those key factors affecting bias. During the statistical analysis, we're trying to use appropriate methods to minimize those potential confounders. We also can use sensitivity analysis to assess the threat of bias. Okay, in the next topic, Suzanne will take a couple of items to the case study for further illustration. Now let's turn over to Suzanne.
Thank you, Junma. Today, I will discuss a case study using a retrospective natural history study in an ultra-rare neurologic disease as an external control. So the overarching objectives of the natural history study and analyses were to inform and support clinical trial design, obtain information on the target population, and to identify potential endpoints for interventional studies. In addition, the objective was to provide data that would serve as a control in subgroups of patients with comparable clinical manifestations and to aid in interpretation of clinical studies with no concurrent randomized control arm. During the planning stages, feedback was received from the FDA, outlining the information to provide regarding the use of the natural history data in lieu of a concurrent control, such that it would be acceptable. Specifically, it was noted that for natural history data to be interpretable, the control patients must be as similar as possible to the population that will receive the experimental therapy with respect to various characteristics, such as their genotype, demographics, baseline functional status, and concomitant therapy, with the exception of receiving the investigational product. In addition, study observations should be performed using the same methodology and timing in the controls and the experimental subjects. The selection of the patient population from the retrospective database involved two steps: the selection of the eligible sites and the identification and selection of eligible patients within each site. This is typical of ultra-rare disease in which available data resides in a handful of centers of excellence worldwide. Once the patient population was identified, the clinical data was abstracted from the collected medical records. The variables selected to be extracted included endpoints that would be compared with the study endpoints. In particular, a validated scale that was validated for application retrospectively to the medical records was applied by medical experts. The retrospective natural history study fell under the regulatory umbrella for an IRB-approved research biorepository. There was no formal standalone study protocol for the data that we were interested in. Therefore, in collaboration with the investigators at the academic institution responsible for the data, a protocol was written collaboratively by clinical operations colleagues at PROMETRIKA to define a sub-study of the observational project, focusing on patients of interest and intended to be used as a historical control for comparison with patients in clinical trials. In addition, in order to derive an estimate of the proportion of untreated subjects that would meet the primary endpoint, detailed specifications were written to facilitate the identification of eligible subjects in the database that would meet the specific inclusion and exclusion criteria used for the clinical study. Furthermore, a formal analysis plan was written by PROMETRIKA with the primary objective of gathering insights into the completeness of data for key outcomes of interest. There are complications in defining time points using retrospectively collected data. In particular, the definition of a baseline encounter needs to be identified. In this case, baseline was identified to be the earliest time point at which a subject met all of the inclusion criteria and none of the exclusion criteria used for the clinical trial. This was accomplished by working backwards through time through the medical records until the criteria were met. This is often difficult due to lack of dates, which is a practical consideration to consider due to privacy concerns. Subsequent encounters also need to be addressed since there's no set visit schedule for when a subject will have an encounter in the medical records. All subsequent encounters were categorized into intervals of time, mimicking the visit schedule of the clinical trial, using that baseline time point as an anchor. The information to be extracted from the natural history database included patient characteristics comprised of demographics and baseline characteristics to be used as candidate variables to match the natural history subjects with those in the clinical trial by employing propensity score-based statistical techniques. In addition, disease characteristics, key findings over time, such as neurologic findings, data on the primary endpoint of interest, and any other relevant available data, such as functional scales or quality of life measures, were also obtained. Referring back to the slide that Jinma showed, the use of external controls is most persuasive when certain criteria are met. In particular, this study was acceptable to the FDA because there is no feasible control. Specifically, due to ethical concerns and significant support for efficacy preclinically, there was no feasible control. A large treatment effect was also expected. In this study, it was expected that only 5% of the untreated compared to 50% of the treated would avoid clinical deterioration over the course of the study timeline. There is also no currently approved treatment for this disease. This disease has a predictable course with a very rapid decline within 2 years among subjects with early onset. The outcome measure was also objective. In this study, the outcome was defined as time to clinical deterioration, as well as change from baseline in the same measure. Finally, this was acceptable because the patients selected would be from a similar population and setting. Due to the rare nature of the disease, most subjects in the clinical trial would be identified from many of the same sites and medical experts that provided data to the retrospective cohort. Finally, in order to mitigate the potential selection bias that can occur in the treated versus untreated group assignment in the primary efficacy comparisons, propensity score, or the predicted probabilities of group assignment, will be estimated for subjects using a logistic regression model with baseline demographic and clinical variables as predictors of group assignment. Propensity scores will be grouped into quartiles and used in analyses as a nominal classification covariate. This study is ongoing and is a work in progress. However, this is the general approach that has been taken thus far, and we anticipate further discussion with the regulators this year. Back to Mignesh.
Thank you, Suzanne. That was a great presentation, and many thanks to Jinma. That was excellent. It was really informative. Thank you so much. Here you have it. We covered the surrogate endpoint development aspects, the external control in deep detail, and also a great example of application of those techniques in a case study. We now will go to question and answers, please. A question for Jinma. Jinma, could you expand a little bit the information about the types of propensity score-based methods and their potential advantages and disadvantages? I know most folks are familiar with propensity matching, and then other methods surrounding propensity scores. Could you elaborate a little bit about that?
Sure. Thank you. That's a good question, Mignesh. We all know that the RCT, the single arm, it's RCT, it's perfectly balanced the treatment assignment, right? For the propensity score-based method, they're trying to mimic the RCT. So that's the first step. What's the propensity score? That is a calculator. Usually we use a logistic regression to estimate the probability or the likelihood of the treatment assignment. So using all those the potential confounders as a predictor. So the dependent variable is the treatment assignment, but the predictor is the confounding potential confounding variables. And then we have this propensity score. So what's the next step? We have a couple of the methods. As you mentioned, we could use the propensity score matching method. So how we're matching it, we could have the propensity score. For example, this patient, they have 0.6, it's a propensity score. We need to look for another control. They also have the 0.6 or very close. We should set up those threshold to find this control. We could use a 1-to-1, we could 1-to-2, this is the matching method. This is a good one. After the matching, the baseline characteristics should be all similar between the two arms. So this is the mimic RCT, the kind of RCT. But this have a little bit limitation for user matching, especially for you in rare disease oncology. The sample size is very small. We're trying to use all patients. We don't want to lose anyone, right? When we're using matching, sometimes it's very difficult to match everyone. Even so the control will probably a big pool that will exclude those patients are not being matched. Even for the case group, some case maybe cannot find any control because this is the prevalence score, it's just cannot find it. So this is the limitation for user matching. We have also another message is a propensity score that's called stratification. Yeah, let's lose up not one-to-one matching. We could categorize those the propensity score. Use a couple of 5 group or 6 group. There's from low to high, there's the propensity score. Then we could use that as a stratified analysis. Just like there's the matching is a 1-to-1, the stratification like by the group is the stratum, right? With this matching in the stratum, with the stratum. And this is all similar. It's idea similar to the matching, but it's a little bit looser strict to the like the matching. We also have, you probably heard a lot, there's a weighting method, still use propensity score based, right? This is the weighting method. One of the most common uses the IPTW. So we know that the two group is not balanced, but it's because whatever it's age, gender, something is not balanced. Then we could use the inverse of propensity score, trying to equalize the two group. So this is a weighting method. Also a couple of other weighting methods I listed there, like a standardized mortality ratio. This is a method of a treatment group just set of 1. It's still the weighting, but the control group, you use the propensity score divided by 1 minus propensity score. So this is called SMR. So another method is called overlapping. This is in the treatment group use 1 minus propensity score, and the control group use the propensity score. So this is all similar technique, but it's different weighting methods. The good thing is the weighting methods, they do not need to exclude any patient. They will use all the patients, all information we used. I hope I already answered your question. Back to you.
Absolutely. And I know that this is a question that is probably on the mind of many in our audience. And I completely agree with everything you said. And thank you so much. That was a wonderful explanation. I appreciate that very much. We have a question from the audience. And specifically, could you comment on the access to outcomes of external control data source to ensure the analysis integrity? So what sort of access and availability, I would imagine, in terms of from the regulator's point of view, when we have clinical trial data, there is validation, verification, there is monitoring, there is all of this that occurs. And if the data sources for an external control don't come from a regimented, let's say, natural history prospective trial data, there may be some issues with establishing integrity of analysis. What are your thoughts on this topic, Jima?
I'm sorry for so for access to the outcomes to the external control, right? Yeah. This is a question is if that some outcome is very easier, like overall survival, right? It's the mortality. It's easy whenever in the single arm trial or external control. It's very easy to define. It's we could easy to establish that to define the outcome. But for some outcome like progression, is like PFS or the response overall ORR objective response. That's in the trial, we certainly have very clear definition to get those outcomes. But for in the external control in the historical data, sometimes this is difficult. But we will use some programming to get this called the real world response or progression. Based on those imaging data, or some it's the biomarker data, trying to get proxy of those outcomes. Yeah.
Agreed. That definitely is a challenge. And I think our audience member, the question is exactly right on. And also to your point, there are endpoints that the validity can be established a lot easier, external endpoints like, for example, mortality versus some that, additional steps will need to be taken. Excellent question. Thank you, Jima. Excellent answer. The next question is on single arm study with external control. How do you define a significant treatment difference? So it's more with respect to how do you, in planning stage, plan for that in sample size and all of that? How do you define that?
Thanks. So this question, yes, in my understanding, this could be similar to the RCT, right? For the RCT one, we calculate the sample size to the power analysis. We already have the hypothesis. What's that? For example, we use the response ratio, the ORR. In the treatment group, we have pushing, for example, we maybe assume it's 60%, but for the control arm, we assume it's around the 30%. That's we already have the assumption that's based on our previous data or based on the literature review. But for the same thing, for external control, the single arm trial, they only have this treatment, but we use the external control. We also have those assumptions, right? We could use the standard of care. What's the ORR for the standard using the standard of care? Based on the literature review, or you based on some other previous data, you could set those the assumption. So based on those assumptions, then you can do the power analysis to calculate the sample size, right? This is the similar, and also this is based on what kind of approach you will use. Use the matching, or you want to use the weighting methods, or this is all could you could apply different the power analysis to estimate the sample size. And once you have this design, then it's the same thing. You can get the p-value to get the confidence interval to to estimate the treatment difference.
Agreed, definitely. And also what I have seen done, and we have done in the past as well, is when we set out to collect data for the external control, in the beginning stages of that, you can essentially do a pilot that you could use for your sample size estimation as
Yes.
well, using the same mechanisms, whichever mechanism you're using to collect your external control data. So as Jima said, depending on how much information is out there in the literature, and what we can use as a starting point, sometimes you may even use the same data collection mechanism to inform you about the sample size. And then from there, you collect data as your external control using that sample size and study design. But I agree, it's not very different from a RCT sample size estimation consideration. And along the same veins, I have a question for Suzanne. What would you say, as someone who has written SAPs for historical control trials and for external control trials in general, what would you say is similar and what would you say is different from an RCT SAP?
Sure. So there are going to be some differences in your statistical analysis plan. Specifically, there should be a section on how the propensity score modeling and the methodology that you're going to use to mimic the randomization, as Jinma alluded to earlier. But aside from that, a lot of the principles that we follow will remain the same. We're going to be pre-specifying the endpoints and the analysis methods, how we're going to handle any missing data, methods to reduce bias, and a section for considerations on multiplicity adjustments.
Agreed. Very well said. And, all those considerations are still at play, as you said. There's a question for Jinma. And the question is, if our endpoint is a responder rate, is that an issue with the external control? And specifically, the question is asking, is it hit or miss if our endpoint is a responder rate?
Yeah, this is a good question. Before I just give example, I did use the RR for the power analysis. But yes, the responder rate, as I mentioned, when we're using historical data, natural history, the responder, because the data is sometimes it's maybe it's missed, underreported, or some not easy to not everything we could capture there. So that's the problem. It is a challenge, but we are trying to, but it's use some. This depends on the study. Some studies are very clear. They have this definition of the responding, or not responding or progression. They have very clear definition by using the biomarker or some of the imaging data. But for some studies, it's very difficult. Yeah, that's certainly the challenging thing. I don't know. Hopefully, I answer your question on that.
I agree. I mean, there are some specific challenges, and also there's some specific challenges with survival analysis as an endpoint in single arm trials compared to an external control. So, but again, in pre-planning, in pre-specification of the analyses, a lot of these can be dealt with and addressed. I would say that it's not necessarily hit or miss to our audience member if it's done properly. Do you agree, Jima?
Yes, I agree. This is my own experience. For example, in using a Flatiron database, right? They certainly have the mortality for OS, overall survival, for sure. There's no question about that. But for the overall response, the Flatiron, they use some programming, get the real world response, the complete response, the partial response. They also have this real world progression data. And it's also this validated. This is good. They could use it from the lung cancer or some other cancer studies. Yeah, just to give more little bit comments for you.
Perfect. Thank you so much. That's wonderful. Thank you, Jinma. Thank you, Suzanne. Many thanks to all of our audience members for attending today. I hope this has been helpful. And I hope that we all, I know that we have learned something today in this 1 hour that we have spent together. I very much appreciate your attendance, and we will follow up with some takeaways from this session for our audience members, including the slide set and other takeaways for you to have and to refer to. Thank you so much again.
Thank you.
Thank you.
Bye.